In an academic world tyrannized by political correctness, even to study the molecular basis of such a characteristic has been something of a taboo. What little we understand about it depends on old studies of mixed-race children, which established that several genes contribute to pigmentation. But our knowledge of other species and the similarity of basic biochemical processes among all mammals suggest a more complicated picture. We know, for instance, that many genes affect coat color in mice, and it is likely that these have direct human equivalents. So far, though, we have managed to identify only 2 genes involved in human pigmentation. The one that, when mutated, causes albinism, and the other, the melanocortin receptor, associated with red hair and a pale, often freckled complexion. The melanocortin receptor gene is variable among Europeans and Asians, but invariant among Africans, suggesting that there has been strong natural selection in Africa against mutations in the gene, i.e., against red-haired, fair-skinned individuals. Albinos who lack pigment altogether occasionally appear today in African populations, probably through de novo mutation, but their acute sensitivity to sunlight puts them at a severe disadvantage. Another morphological trait likely determined by natural selection is body shape. In hot climates where dissipating body heat is a priority, 2 basic types have evolved. The nilotic form, represented by the East African Maasai, is tall and slender, maximizing the surface area to volume ratio and thus facilitating heat loss. The pygmy form, on the other hand, though still lightly built, is very short. In this case, a physically strenuous hunter-gatherer lifestyle has selected for small size to minimize the energy expended in movement. Why lug a big body around to look for food? In high latitudes, by contrast, selection has favored body forms that promote heat retention, those with the lower ratios of surface area to volume. Neanderthals from northern Europe were therefore heavily built, and so too, on average, are today's inhabitants of the same boreal climes. Some of the variation in athletic performance we see among groups is presumably attributable to these body form differences. It should come as no surprise that in the high jump, for instance, a tall nilotic body is better adapted than a short, robust one. If there is a trait whose distribution among human populations is hard to fathom, it is lactose intolerance. Mammalian milk, including the human variety, is rich in a sugar called lactose, and newborn mammals typically produce a special enzyme, lactase, to break it down in the intestine. Upon weaning, however, most mammals, including humans, at least most Africans, Native Americans, and Asians, stop making lactase, and so as adults cannot digest lactose. Lactose intolerance means that drinking a glass of milk can have unpleasant consequences, including diarrhea, gas, and abdominal bloating. Most Caucasians and the members of a few other groups, on the other hand, continue to produce lactase throughout their lives, and can therefore handle a lifelong dairy diet. The explanation has been advanced that lactose tolerance evolved in those groups historically most dependent upon dairy products, but the pattern of the trait is by no means fully convincing. There are, for example, groups of Central Asian animal herders, cheese for everyone, who are lactose intolerant. And despite belonging to an ethnic group that is typically lactose tolerant, I am intolerant. If natural selection had favored tolerance in a particular group, why would it leave its job undone? The most compelling evidence yet in support of the standard explanation is the presence of lactose tolerance in African groups traditionally associated with livestock. We may never fully understand the adaptive dimension of this trait, but molecular biologists working on a Finnish population have recently identified the mutation responsible for it. And so, while we are by no means fighting a killer here, it is now possible with a simple genetic test to determine whether a newborn will grow up to face a choice between ice cream deprivation and chronic gastric cramps. More interesting than the relatively few differences we see among the races is what we all have in common, what it is that makes us so different from our closest relatives. As we have seen, our lineal split from the chimpanzee about 5 million years ago has barely given us enough time apart to evolve a 1% genetic difference. But in that 1% lie the critical mutations that make us the remarkable thinking, speaking creatures we are. It may be debated whether other species possess some limited form of consciousness, but clearly none of them has produced a Leonardo da Vinci or a Francis Crick. The chromosomes of humans and chimpanzees are very similar. Chimpanzees, however, have 24 pairs, whereas we have 23. It turns out that our chromosome 2 was produced by the fusion of 2 chimpanzee chromosomes. There are also differences in the human and chimpanzee versions of chromosomes 9, bigger in humans, and 12, bigger in chimpanzees, and several examples of inversions, or flips, within chromosomes that differ in humans and chimpanzees. Now, whether these chromosomal differences will prove significant is hard to say. The relative merits are not much clearer at the biochemical level, where so far we know of only 2 differences between humans and chimpanzees. Difference 1. In both species, a sugar molecule called sialic acid appears on the outside of every cell. But while the molecule is subtly modified in chimpanzees through the action of an enzyme, in humans, the gene encoding that enzyme is always mutated. No enzyme is produced, and human cell surface sialic acid is unmodified. We have no clue at all as to whether this is significant. Difference 2. This one, discovered in 2002 by Svante Pääbo's group, is more suggestive. A difference in FOXP2, a gene known to be involved somehow in human language. Because mutations in the human version have been found to cause linguistic impairment, FOXP2 has been misleadingly dubbed by the media as the grammar gene. Out of a chain running 715 amino acids, just 2 changes distinguish humans from chimpanzees and gorillas, whose FOXP2 proteins are identical. In fact, these amino acids are identical in all mammals tested, except for humans. Moreover, statistical analysis of the pattern of DNA variation in and around the gene suggests that natural selection may have had a role in shaping the protein during human evolution. It is therefore tempting, but premature, to suggest that FOXP2 is the evolutionary equivalent of a smoking gun, a glimpse of a critical step in the origin of language.